Hello, thank you all for joining us. I'm Matt Hubeck, the moderator for today's webinar. If during the presentation you have any viewing difficulties, go ahead and hit F5 on your keyboard to refresh your browser window. Feel free to submit questions during the presentation. We will do our best to address your questions in a Q&A session with the time we have left. If we do not get to your question, we'll do our best to respond via email. With that, I'll hand things over to Peter Brescia.
Hello, thank you all for joining us today. As you can see from the title slide, today I'll be discussing microvolume analysis using a microplate reader, and I'll discuss quantification of DNA, RNA, protein, as well as some cell work. With that, I'd like to start off by presenting a poll question to get a feel for what you all are working with today. So I will go ahead and present that to you now. Did you push that slide, please? Push slide, please. Next slide. There we go. Sorry about the delay there. So the question is, what sample types are you typically processing that would benefit from the ability to utilize as little as 2 microliters for accurate quantification, and for what downstream applications are they used for? A, gel separation, B, sequencing, C, PCR, D, molecular diagnostics, E, cDNA synthesis, F, gene expression analysis. I'll give you a moment to answer the question. Slide, please. Great. Okay, so looks like we have a variety of answers here, so that's good. Folks are working in all those areas. Hopefully what I'm about to present to you today will cover some useful techniques that you can use for all of those processes. I'll provide you a brief outline at this point. First, I'll give a background discussion of quantification of DNA, RNA, and protein using absorbance methods, and then I'll move on to a discussion of microvolume analysis of nucleic acids and proteins, quantification of those molecules, as well as some purity assessment techniques for nucleic acids in combination with spectral scanning. And then I'll provide an application on labeling efficiency, and that'll provide you with a practical application. And then we'll move on to look at some in situ microvolume assays. This will allow the ability to extend the dynamic range of quantification in microvolume. And we'll discuss the pico-green assay as well as BCA assay for protein analysis. And then I'll round out the talk with a discussion on microvolume analysis of cell growth. So starting with background, first I'd like to discuss absorbance spectroscopy, and many of you will be familiar with this. We'll have a quick discussion here on the Beer-Lambert law. Most folks are familiar with the use of UV spectroscopy, and traditionally that has relied on the use of a 1 centimeter vessel. And that simplifies the Beer-Lambert law here, where we're looking at incident light here on the top, and then we're looking at a ratio to the incident of light that's actually transmitted once it's passed through the sample. And by the use of a 1 centimeter vessel, we can simplify this equation, and using the molar absorptivity, we can calculate the concentration here simply by dividing the absorbance by that molar absorptivity. Of course, that molar absorptivity is specific to the biomolecule that you're looking at. Traditionally, this has required a fairly large sample volume. Most of the cuvettes require at least 1 to 2 mils of sample, and the sample to be fairly dilute given the 1 centimeter path length. However, those dilution steps can be eliminated by reducing the path length, and that has led to the use of microvolume analysis. And that's what we're going to discuss today. So I'd like to introduce you to microvolume analysis here at BioTek using our instrumentation, as well as discuss some other instrumentation. So if we think about the typical workflows that apparently most folks are using based on the poll question, we start with some sort of sample here, and this can be tissue, body fluids, or cell culture, which I'm sure most folks are using as well. And typically that process starts with isolation of either DNA, RNA, or protein samples, and we'll need to quantify those for efficient downstream workflows, such as would be required for quantitative PCR or quantitative RT-PCR. So essentially all these paths lead through quantification, and we need to have accurate quantification to be able to work through these different workflows. So microvolume analysis, here I'll introduce you to the microvolume microplate that BioTek offers, and I'll do a short comparison between the other methods that are currently used, such as 1 centimeter path length vessels, as well as microplate vessels, which most folks are probably familiar with as well. And a lot of this work was done on a standard absorbance microplate reader. So I think it's important to understand the differences between the three methods and what makes them the positives and negatives of using each type of vessel. If we first think about the standard BioCell, which is a 1 centimeter path length vessel, or a standard cuvette, we have a fixed surface both at the top as well as the bottom, which defines our path length of 1 centimeter. If we then think about a microplate, which has one fixed surface down at the bottom, the path length will vary depending on the volume, and it can range anywhere from 5 millimeters to about 10 millimeters. And it's important that we realize the limitations of that. We need to accurately define that path length to be able to define or to utilize the Beer-Lambert law. BioTek software, the Gen 5 software, does have the ability to determine that path length using path length correction, and other methods would require that you know the volume and the measurements of the vessel, and you can calculate the path length. A little more tedious working that way. With the Take3 microplate over here on the left, we have two versions of that right now, but essentially they both work the same way. It opens up sort of like a clamshell, but when it's closed with a sample in there, what you'll see is that a column is formed between the top and bottom surfaces of the slides, and that produces a nominal 0.5 millimeter path length. This allows you to use samples as small as 2 microliters and to accurately quantify those. And as you can see, there's multiple spots for reading. In the standard Take3, there's 16 spots, whereas in the Take3 Trio, up to 48 samples can be loaded. And again, those can be as low as 2 microliters. Now I'd like you to look at a
Take3 is a
quick
simple
clip
device
here
allowing
that
rapid
introduces the
quantification of
Take3.
multiple samples using a multi or single channel pipetter. Once loaded, the Take3 creates a nominal 0.5 millimeter path length capable of being read on a microplate reader. Up to 16 2 microliter samples can be loaded onto the microspots. The Teflon coating provides a hydrophobic surface resulting in droplet formation. Once the samples are loaded and the cover closed, samples are ready for measurement. After reading, simply wipe the samples off with a laboratory wipe or recover by pipetting. The Take3 is flexible, containing additional locations for 1 centimeter path length vessels, such as a standard cuvette or a BioCell.
So now I'd like to introduce you to some of the data the analytical performance data of the Take3 plate. We'll start with protein quantification. This data here was generated using a BSA standard protein as essentially a calibration tool, and to determine not only the accuracy compared to a 1 centimeter path length vessel, but also in comparison to what most folks are probably familiar with, the nanodrop as another microvolume analysis tool. If you look at the graph on the left, what you'll see is a straight line analysis, and the y-axis represents microvolume determined BSA concentrations, and these are in milligrams per mil, with the insert being the lower end of that range that was tested. And then the x-axis is biocell or 1 centimeter path length determined concentrations. And what you'll notice is that we have a near perfect fit, and it compares well also with the nanodrop, and we've gone down to essentially very low milligram per mil or high microgram per mil concentrations down into the hundreds of micrograms per mil. And again, this was done with 2 microliters of sample. Now, if you look at the bar charts over on the right, you'll notice in the top we have accuracy versus the 1 centimeter path length, and in blue is nanodrop, red is Epoch Take3 comparison, again across the range of protein concentrations from 50 mg per mil essentially down to a couple of hundred micrograms per mil. And you can see we have a bit of an improved performance relative to the nanodrop, with most of the percent accuracies being below 5%. If you now look at the bottom bar chart, you'll see CVs and replicates. These are either 10 or 16 replicates for the nanodrop, Epoch Take3 respectively, and you can see we have excellent CVs, generally again below 5% until you get to the very low microgram or 200 microgram per mil concentration of BSA. Next I'll show you some data with DNA. Again, over on the left, the graph over here shows microvolume determined BSA concentrations again on the y-axis compared to 1 centimeter vessel concentration determinations on the x-axis, and nanodrop is on here as well for comparison. And again, you can see a near perfect correlation between the 1 centimeter vessel measurements and those determined at a microvolume. And furthermore, you can see that over here on the bar chart on the right that we've had a look at double-stranded DNA purified from CHO cells using a QIAGEN All-Prep DNA RNA kit. And all of those concentrations here we're looking at determinations from on the far left would be the Epoch Take3 2 microliter microvolume measurements. And then we compare that with microplate here in the second column versus a 1 centimeter measurement taken here in the third column, and then cuvette, and finally nanodrop. And all of these, again, CVs are about 1%, and all are within 2% accuracy compared to the 1 centimeter measurements. So the take-home point here is that we have equivalent performance across all 3 levels of analysis, the microplate, microvolume, and cuvette, and all are equivalent to those determinants by the nanodrop as well. Lastly, I'd like to show you a slide, essentially the same type of data, this time RNA. Again, we can see equivalent performance, both the straight line analysis, again, near perfect correlation between 1 centimeter determined concentrations and microvolume, and as well as, again, CVs below 1% and essentially equal performance across all of the vessel types tested, as well as the nanodrop. Another aspect of quantification, as well as being able to determine the concentration, is the ability to know that what we're reading is truly absorbance from a purified sample. Most commonly, folks refer to this or purity assessment as using an A260, A280 ratio. And if we look at the average double-stranded DNA coefficient at 260 nanometers, it's about 0.2 nanogram microliter per centimeter, and 280, 0.1. And so that gives us an expected ratio of 2. And most folks are typically looking for a pure dsDNA sample to have a ratio between 1.8 and 2. And similarly, if we look at the coefficients, average extinction coefficients for protein, here we would expect a pure sample to have a ratio of about 0.57. And so typically when folks read absorbance and look for quantification of those samples, they'll run several wavelengths, often a reference wavelength as well at 320, and then they will determine this ratio to make a decision whether or not the sample is pure. However, it's not always the case that we can tell whether or not a sample is pure, and I'll discuss some additional methods using spectral scanning and show some evidence of why we might want to use some other methods as well as standard A260, A280 ratio. First, if we take a look at just the spectral data of a pure versus complex samples, if we start here on, with the graph on the left and we look at increasing concentrations of pure double-stranded DNA, we'll notice that indeed we do have an absorbance peak at 260, which increases with increasing concentration. Now if we look at a complex sample, as well as purified DNA and protein, what we'll notice is that the sample contributes, especially the DNA sample, will contribute quite a bit due to its absorbance peak at 260, whereas protein, its major contribution is at 280. And so, of course, that's why we quantify protein at 280 and DNA at 260, as well as calculate that ratio. What we'll notice here with the essentially the data that's plotted that runs between the two, this is a complex mixture of 60% DNA and 40% protein. And what you notice is that it takes quite a bit of protein in that sample to begin to see a decrease in absorbance at that at the 260 wavelength. And I'll show you some further data here, looking at those ratios. Oh, there we go. So now if we look at some typical A280 ratios of complex mixtures, now if we look at the graph at the left here, what we're looking at is decreasing percentages of DNA with increasing concentrations of protein, again, as a percentage. And I'm not sure if you folks can read those or not, but it starts at 100% DNA up here at the top, and then we have the next concentration would be 60% DNA, 40% protein, and then the inverse for the next 40% DNA, 60% protein, and so forth. And when we get down towards the bottom, these are very small relative percentages of DNA in the 2 to 4% range with very high protein concentrations. And again, you'll notice that we see a decreasing absorbance peak at 260. However, you'll also note that we see very little change at 280, which can make the determination of the A260, A280 ratio for purity assessment of protein somewhat difficult. So now if we switch our view to the graph on the right, now we've plotted a spectral scan of absorbance from 240 nanometers to 290 nanometers and plotted those as a ratio all divided by 280 nanometers. And so now we're looking at a ratio over on the y-axis. And what you can see, which is expected for DNA, we have a peak ratio of about 1.8, 1.85 for a pure double-stranded DNA sample. And then, of course, for protein, again, you can see very minimal A260 absorbance And then if we look at the A280, 260 ratio here, we're looking at about, should be about 5 or about 8 for ratio. So a little bit higher than what we might expect for an ideal, but certainly that's will change from sample to sample. Now if we look at our complex sample, and in this sample we have a 60% DNA sample and 40% protein weight, what we see is that it really takes a tremendous amount of protein. Here we're showing 40% protein contamination, and we're still seeing a ratio of about 1.7 or 1.75. And so I guess the take-home message from this slide is that it takes quite a bit of protein contamination in your sample before you'll actually realize that on a A260, A280 ratio determination. If we look at this data a little bit differently here now, what we're looking at is on the left a graph of how that ratio changes with the increasing concentration of protein and decreasing concentrations of DNA. And again, as I mentioned, 40% of protein contamination results in a very small shift in that A280, A260, A280 ratio. What's more important is if we take a look at the table over here, we can start to see what kind of effect that has on the accuracy of our DNA and protein determinations. And we can see with approximately 40% protein contamination, it doesn't really affect our DNA accuracy that much, 10%. That's a very high percentage of protein contamination, not something you would typically see, or hopefully you wouldn't typically see in your samples. Whereas if you look at your at the lower end of your protein contamination or minimal amount of contamination with DNA, this down here, if we look at this 15.7% accuracy, that is a very, very slight DNA contamination of about 1.8% in our samples. So the take-home message here is that it doesn't really take very much DNA to throw off the accuracy of our protein quantification, whereas it would take quite a bit of protein contamination to throw off our accuracy of our DNA quantification. And both of these effects, contamination in general, can have a tremendous effect on our downstream workflows. We know that contamination can certainly change the efficiency of many of our reactions, such as PCR, quantitative PCR reactions. So if we think about some of the other contaminants, this question comes up quite a bit. So, some of the typical things that you might use to purify DNA, such as guanidine hydrochloride or phenol, what sort of effects do those have on quantification, and can you see those using a 260-280 ratio? So we get this question quite a bit, and so here we've taken a look at, if you look at the graph over on the left, you'll see we've plotted several different contamination quantities of guanidine hydrochloride ranging from 0.5% volume to 10%. 10% certainly on the high side, but you might expect to see something, if you have contamination down in the 0.5 or 0.1 to 0.5 range. And what you can see is that it only takes a very little bit of contamination here to throw off this absorbance reading quite a bit. And then again, if we take a look at the accuracy calculated, you'll see that 0.5% we're already looking at a minus 12% accuracy. So in this case, we'd be underestimating the amount of DNA that's in our sample by about 12%. And if you take a look at the A260, A280 ratio, you can see that it does not really change all the way up to 10%. So this probably isn't the best method for determination of guanidine hydrochloride. There might be other tools that would be more suited to this type of determination. Next, if we take a look at another common contaminant that's found, phenol chloroform extraction of DNA is a very common method. Again, if we take a look here, first at the spectral scan of phenol contamination, here we've plotted 0.2% phenol up to 1% phenol contamination, volume again. Now in this instance, we can certainly see visually a shift in the peak absorbance from 260 to approximately 270 or 268. This would certainly provide much more useful information to a user to run a spectral scan if phenol contamination is thought to be happening. And you can see that there is significant differences in the 260-280 ratio calculated. Here we're starting at about 1.9, and we're already out of the range that we would typically expect to see for pure DNA once we hit about 0.2% contamination. And again, we can see there's a very significant impact, again, due to that decrease in the peak absorbance at 260, where here we're seeing an increase. We're seeing a very large increase in the calculated DNA concentration here, about an increase of 41% accuracy versus 1 centimeter path length vessel calculations. So take-home message from this slide is, yes, phenol can probably be determined even at very low quantities and easily determined that it's phenol, not only by looking at the mainly by looking at the spectral scan, but we will certainly be clued into a contamination simply by looking at an A260-280 ratio. I'd like to now change gears here slightly and just discuss a practical application, one that we're currently working on here at BioTek. And this has really been an excellent use for the microvolume analysis. And I don't really want to talk much about the details of alpha ELISA. I'm sure many folks are already familiar with this type of technology, proximity assay type technology. What I'd like to point out, though, is in this case, labeling of an antibody. And whenever we do that type of labeling, we need to quantify not only how much product we have at the end of the labeling, but we also need to determine labeling efficiency of that product for use in our downstream application. For this particular alpha ELISA assay, we needed to biotinylate our antibody drug, and we used a reagent called Chromolink biotinylation reagent. It's a really nice, quick method for labeling. And this particular label has a chromophore on it that allows us to easily quantify the substitution ratio, or how many moles essentially of label we have on our protein. And at the same time, we're able to read our absorbance at A280, which is not interfered with by the labeling reagent, and we can determine our protein recovery. And so what we've done here, if we take a look at the graph down on the left, is we've run a spectral scan, and these are duplicate measurements done with 2 microliters of sample. And I guess this is a good time to mention one of the main reasons why we like to do work at microvolume volumes is due to the reagents themselves being quite valuable, and or the antibodies or proteins or DNA samples that we're working with. And this was one of those cases where we essentially end up with 50 microliters of purified biotinylated reagent that we need to quantify, and we certainly don't want to put 50 microliters or all of the sample into a large vessel and then try to recover it and hope that we haven't contaminated it and so forth. In this particular instance, I did do two microliter samples, ran a spectral scan on those, and then I was able to recover those as well by pipetting them off of the slide and putting them back into my reagent vessel. And what we're looking for here is a typical ratio of about 3 to 8, and it might be hard for you all to see, but we ended up with a ratio of about 5.42 and corrected protein concentration of about 0.21 mg per mil. And this is enough to run a sufficient number of assay data points for what we're looking for. So this was a really quick, easy method to quantify not only our recovery as well as our labeling efficiency. So as I mentioned earlier, I wanted to also introduce you to a few methods to increase the dynamic range. We know that we're somewhat limited in the ability to quantify samples at very, very low concentrations. I also touched on the ability to measure samples at very high concentrations using the shorter path lengths, but the opposite is also true. It's more difficult to quantify low concentration samples using a shorter path length. So we might need to use some enhancement reagents to increase the dynamic range if we still want to conserve sample. So I'll introduce you first to the in situ picogreen assay that we developed here. It's a modified version of the standard assay that folks typically run in a cuvette or microplate-based application, and all the details won't be presented here today, but they are available as a downloadable application note from our website. Excuse me. So if we take a look at the in situ protocol over here on the left, it's pretty straightforward. What we do is we add our sample, DNA, protein, it'll be the same regardless of what the protocol is, directly to our slide. And then we'll next add 2 microliters of our working reagent, and then we'll close the lid and incubate right on the slide, and then we'll pop the Take3 into a microplate reader and read the absorbance or fluorescence, depending on what the assay is. So it's here we can see the working range of this particular assay. As we developed it, the range of our standards that we typically run are 10 to 1,000 nanograms per mil. That allows us to gain several orders of magnitude in sensitivity relative to the standard absorbance reads at A260. This particular protocol is simpler and faster than the standard protocol in that our incubation time is only 5 minutes, and of course the reagent and sample savings kind of are spelled out as well. And if we take a look at this graph, we can see down here on the bottom, I'm not sure if you can read it, but we've read this on both a Synergy H4 and a Synergy HT. So we did comparison between using spectral filters, which we know allow higher energy through, versus monochrometers, and you can see that we get excellent correlation regardless of the method that we use. So this is a flexible technology, can be used across several of our instruments, including hybrid instruments that have fluorescence-based monochrometers. Next, I'd like to spend a couple minutes discussing a second assay that we've adapted to microvolume, and this is the BCA assay. I'm sure most folks have probably used this if they've worked with proteins, and essentially this reaction requires the peptide bonds to reduce the copper, which then forms a complex that absorbs at 562 nanometers. And so again, we've changed the protocol. We've adapted it to work in a microvolume, and part of that was changing the ratio of reagents, and we've changed that ratio. The common ratio is 20:1. We went to a 1:1 ratio, and again, we're running this in situ, so we're adding sample and reagent directly to the slide and allowing it to incubate. And what you can see is that we, over here on the bottom left, by looking at this chart, the in situ analysis provides a much higher signal compared to the standard 20:1 incubation. And this allows us to not only run this in microvolume format, but obtain results at much lower concentrations of protein, or determine concentrations much lower. Over here on the right This assay is compared to the nanodrop, and again, we can see excellent correlation in both microvolume assays. In fact, if you look at the inset, you'll see a little bit better performance on the Take3 system using the Epoch reader than the nanodrop, which the nanodrop tends to be a little bit low, in fact going negative at the very low end. So this is useful. Again, you can gain an order of magnitude of sensitivity. We're looking again down in the very, we're getting now down into the 10 micrograms per mil range. And again, it's much faster and simpler than the standard protocol. This can be run in about 25 minutes, and sample and reagent is about 20 times less than running in a standard either cuvette or microplate-based assay. Lastly, as I mentioned, I'd like to discuss some cell growth, monitoring of cell growth in a microvolume format. Again, the value here is that if you're doing screening of multiple cultures or multiple clones, you can do it in a much smaller volume. Again, we're going to be working at 2 microliters here. So a little background on turbidity measurements. Typically, these are methods that are important. This method is used to look at several different cell culture types, be it bacteria, yeast, or mammalian cells. It's an alternative method to counting on the hemocytometer. It's much quicker, and it's an accurate method to quantify cell mass, and we're going to need to do that again for multiple downstream processes to ensure that we have either the correct biocell mass to move forward with production of an antibody or biologic or protein. It's also used for quantification of yeast strains in alcoholic beverage production, as well as what we do here quite a bit is in vitro model systems for drug discovery and toxicology studies. So turbidity is common. It's a rapid method that's used, and essentially what we're doing is we're looking at optical density at 600 nanometers. It's important to recall that this is not an absorbance protocol, so it does not follow Beer's law, and it will require a calibration curve that's specific for each cell type that you're interested in quantifying. And one limitation that I'll mention is the inability to determine whether the cells are alive or dead, and so that can be an issue in some cases. So you should keep that in mind as well. Okay, so as I mentioned, specific to each cell line, you will expect different absorbance readings. And so, not absorbance, optical density measurements. And so if we take a look here, I'm showing you several different cell lines and their calibration curves. And again, what we're looking at is cell number versus optical density at 600 nanometers. And what you can see is the large squares will represent about 2,000 cells. In this image, top left, these are Dowdy cells, which are peripheral blood cell line that we work with quite often here. So having this calibration curve allows us to do a quick absorbance measurement. Again, all these measurements are done using 2 microliters of sample. So depending on whether you're growing your cells in a microplate, in a microplate reader, or in a cell culture, it allows you to conserve the amount of cell culture that you need to remove to take these measurements. And it allows you to rapidly assess cell growth over time. Next, graph over yeast cells here. These are, again, easily quantifiable using absorbance 600, and you can see we have a very linear response. In this case, that's not always the case. If we look now down on the lower left, you'll see bacterial cells, JM109. Essentially, we get a bit of a curve here that's outside of linear response, and that's typical. Again, you'll see that with HepG2 cells, which are a cell line, immortal cell line, cancer cell line, and again, all of these we can run a calibration curve. Once we have that calibration curve, then we can simply take an absorbance measurement at 600, and then we can utilize our curves to determine what the concentration is of that culture at any time. Here I've just shown an image. This is a bright field image of 2,000 Dowdy cells. So this is what it actually looks like with a 2 millimeter, within the 2 millimeter microspot of the Take3. And this will yield an optical density of about 4 milliOD. And this is about 0.5 millimeters, the depth of the image. So some of these cells are in focus. Of course, this isn't all the cells, so they're not all in focus. And finally, I'd like to show you some data. This is a standard growth curve, bacterial curve, and what we're showing here is a comparison of 1 centimeter path length measurements. So if you were to measure half a millimeter or half a ml of the sample in a 1 centimeter cuvette, or I'm sorry, 2 mils of the sample in a standard cuvette versus 2 microliters of sample. Oh, I'm sorry, actually, those are the wrong units. So that's 2 microliter sample on the Take3 versus half a mil in a low volume cuvette. And you can see we have excellent correlation. You'll notice that these have the absorbance optical density values on the right-hand y-axis have not been path length corrected, so these are the actual values you would expect excuse me, to see. And that's the end of the presentation today. I'd like to thank you all for joining us today, and we'll take some questions at this point. I've got several questions here, so let me pick one here. See, the first question is this microvolume analysis conducted with a fluorescent dye, or is it simply the OD absorbance? So that's going to depend, of course, on what assay we're looking at. Most of the work that I presented to you today was simple absorbance measurements at either 260 or 280 nanometers until we get into the in situ type assays. Of course, one of those used a colorimetric dye for the BCA assay, whereas the Pico Green assay does rely on a fluorescent dye. Okay, let's pick, okay. Next question is, what is your experience with plants contaminants in DNA as polyphenolics and polysaccharides, starch, oil? That's a good question. I haven't worked very much with plants and contaminants in DNA. That would be interesting to take a look at, I think, as we did with the phenol and the guanidine hydrochloride. It'd probably be worth taking a look at at spiking samples with those and see what kind of spectral analysis we get first of all, and see if that is going to be a problem. Depending on what your downstream workflow is, obviously whether or not those are going to interfere with your assay. It might be as simple as not using absorbance and using a chelating dye such as the Pico Green, which is much less or much more tolerant to contaminants and accurately quantifying your sample. Let's see. I think we have time for a couple more here. Oh, not that one. The next question is, what is your experience with total serum plasma protein determinations using the Synergy 4? That's a good question. We've actually had some questions like that come in from customers, and it depends on what your sample is, of course. The total serum, sometimes you're going to have to dilute that in a lot of instances to be able to read it, just because of the, well, it depends on what vessel you read it in. The microvolume analysis is probably a good way to go for this, given the quantity of proteins that are found in those types of samples, and the microvolume analysis having the smaller path length would probably help out there. I haven't tried it here, so I don't know, but I do know, several customers have tried it, and they were having some issues with some of the contaminants that are in there and some of the buffers that are used. RIPA buffer, I think, is used in some of those, and that does contain a detergent which can interfere with quantification. So again, it comes down to a contaminant issue. Which leads to the next question. We are experiencing high variations in protein quantification using Take3 Epoch, getting negative values, and do you think the buffer RIPA used for diluting proteins would be the problem? And indeed, that probably is causing some interference, especially if you're using that as a blank. You're going to have very high absorbance blanking values that will, give you negative values once you quantify your protein, depending on what the concentration of the buffer is in that sample as well. But certainly, probably need to do a buffer exchange into a buffer that does not have a detergent that's going to absorb in that range, or use a BCA assay. That would be the other way to go there. Let's see. So we have a question. The nanodrop measures 230 nanometer reading also to determine purity of RNA. Does Take3 plate have the same features? Within Gen 5, you can certainly control what reference value or what reference wavelength you want to use. So you can certainly do a 260-230 ratio. I didn't touch on that here, but that is certainly something that can be determined, as well as using a spectral scan of your protein or of your RNA samples as well, to get as much information as you can, and then you can certainly use other reference values as well. The next question, the growth curve data on the last slide, is this live kinetic settings data on BioTek plate reader, or samples were loaded externally on the microspots? These were samples that were taken out of the culture at time points and then loaded on the microplate. I guess that's one disadvantage of using the microspots, is that you certainly can't do a kinetic read on that small of a quantity. 2 microliters will evaporate quite quickly, so that's not something that you can do. You could, however, use a standard microplate and run growth kinetics in a microplate if you have a suitable seal on the plate, which they do have optically clear plate seals that you could use. Probably want to do that in a 96 well or lower density, so that you have sufficient volume, depending on how long you need to run that kinetic readout. Next question, is there a method to fluorescently quantitate glucose using a microvolume assay feature? That's an excellent question. We haven't tried that here yet. We do have an app note on fluorescent quantitation of glucose, but we haven't tried it in a microvolume, so that might be worth looking at in the future as well. Thank you for that question. Next question is, the concentration I usually have are around 1 microgram per microliter, assessed by Bradford method using Epoch. Is it too little to quantify using Take3, considering the noise of RIPA? I'm not sure about that. It would depend on the RIPA buffer. I haven't used that here, so that's a good question. It's worth a try. I mean, I would certainly give it a try and see if you can get accurate determinations, as compared to your Bradford method. And certainly that could save you some time if it does work, but we have not tried that here. Next question is, can you please say more about path length correction procedure? It seems to be based on DI water and then used for other samples of the same volumes. Is that correct? That's not entirely true. It is based on water. However, it works on any sample that is aqueous. Essentially, the way that it works is we take the software and reader will take measurements at 900 nanometers and 977 nanometers, which is the absorbance peak of water. And then it utilizes that absorbance peak to determine the path length of that sample. So you do not need to necessarily do that on a well, on a reference well. You can do it right on your sample well as long as it's aqueous based. How do you generate a calibration for the bacterial growth curve? What we did for that was we did a serial dilution and plated out the bacterial culture, and we counted colony forming units, and then we back calculated using the dilution factors to determine how many cells were in that culture, and the calibration curves that you saw were all serial dilutions of a top concentration, which we either counted for the mammalian cells or the yeast cells on a hemocytometer, or used the colony forming units approach for bacteria. It looks like we're I have time for maybe one more question here. Let's see. is it possible to use one calibration curve for quantifying the amount of cells in different days? Yes, you should be able to use a calibration curve. The optical density at 600 should not change for that particular culture unless you're changing the media that the cells are in. Then you might get a slightly different value. However, as long as you're continuing the same culturing process, once you have your calibration curve, you should be able to use that from day to day. And one last one. Please confirm Take3 works in fluorescent assays in addition to absorbance assays. Yes, some of the early Take3 plates that went out did have white slides. So if you already have a Take3 plate, just check to see that it has a black coating on the slides where the microspots are not. And the black coating works significantly better than the white coating. And so if you're purchasing a new Take3 or interested in purchasing a new Take3 or Take3 Trio, those will come with black slides, which work fine for fluorescent type assays. And again, we have several application notes that are available on the website where we've done work with fluorescence on the Take3 and microvolume. Well, I'd like to thank you all for coming today, and we have one last note we'd like to pass on to you.
Okay, thanks for joining us. I've been made aware that the PDF slides don't exactly match the presentation, and they're from a different webinar that we put on. Excuse me. So that will be updated momentarily, so you will have the ability to download the slides via PDF. I just wanted you to go ahead and check out our website and blog. Like us on Facebook, and go ahead and follow us on Twitter if you want to hear more about biotech webinars or learn about our products and how they can help you with your research. Thank you very much for joining us today.